Hello, my name is Julian Moore. I am a junior biology major here at Oglethorpe University. As I'm positioning myself in general medicine, I realized that there's a need for a psychological evaluation for anxiety and depression response. And so I've always been interested in the inner workings of the human brain and how it operates and functions. And so I was particularly fascinated with different emotions such as depression, anxiety, and stress. And so, after reflecting about this past year and a half with the pandemic, I wanted to know why humans experience these certain emotions and why do we perceive them a certain way, and why are certain people able to handle certain situations and handle certain depression and anxiety uh, better compared to others. And so, that's what I'm going to be talking about for the remainder of the talk, identifying novel anxiety and depression receptors in mice. So here are some alarming depression and anxiety data due to the pandemic. And so you can see here from January of 2019 to December of 2020 that there was a massive increase in the symptoms of anxiety disorders and symptoms of depressive disorders, and both of them combined. So this is on the right a chart of percentage of U.S. adults showing these symptoms. So you can see for symptoms of anxiety disorders, it went from 8.2% in January of 2019 all the way up to nearly 37% in December. So just in a little more than a year, these depressive symptoms multiply significantly. And so here on the left, this chart, you can see that symptoms of anxiety and depressive disorders um, is at an alarming high rate at about 36%. And so worldwide, depression will have a lifetime occurrence of about 8 to 12% in all individuals. And so in the US alone, about 30% of all adults will experience some form of anxiety disorder at some point in their lives. And so stressful events like the pandemic can only worsen these disorders in individuals. And so the walkthrough for the remainder of my talk, so I'll be defining anxiety and depression. So what do these look like in individuals? I'll be talking about identifying the major anxiety and depressive receptors. So there are three major receptors that researchers identified. And so then I'll be talking about the specific tests of anxiety and depression that were used. I'll be talking about a forced swim test, a tail suspension test, and a light dark box paradigm. And then at the end, I'll talk about the bigger picture um, of anxiety and depression. So first, I'm going to start off by operationally defining anxiety and depression. So these two terms, anxiety and depression, are two common diseases that are often mistakenly used synonymously, but they are different in their own respective fields. And so you'll hear people possibly using these two terms interchangeably in sentences, but they are different. And so we need to study both of them and produce models that embrace and leverage our understanding of both the genetical, cellular, and physiological responses so that we can produce useful solutions to help treat these disorders. And so for the remainder of the talk, I'll talk about the developmental models to address anxiety and depression in mice. So here you are able to see the symptoms of depression and how they can be modeled in mice. And so here first are the symptoms I'm showing you. Um, Fatigue or loss of energy, and then large changes in appetite or weight gain. But there are some limitations of using a mouse as a model organism, such as thoughts of death or suicide. These are kind of man-made emotions that can't really be quantified in a model organism. So depression is fairly easy to model in mice, and so there are some tools that are used to help quantify depression, some of them being the Diagnostic and Statistical Manual of the American Psychiatric Association. That's one of the tools used to quantify depression, and another one being the International Classification of Diseases at the World Health Organization. So depression is common, as you seen in the previous slide, talking about the increase of depression and anxiety in the United States in adults. And so here you can see how these different symptoms are modeled in mice. So large changes in appetite, you can see how that can be fairly modeled in mice, how there's an abnormal loss in body weight after exposure to chronic stressors. And so compared to anxiety, as you'll see in a little bit, there are more limitations in depression compared to anxiety. So recurring thoughts of death or suicide, that really can be modeled in mice, as well as the feeling of worthlessness or inappropriate guilt. But mice are good model organisms for humans because they provide markers to study cognitive diseases in humans. And so here you can see that anxiety um, is an adaptive response to stressors. And so here are some symptoms that can be modeled in mice, so such as sleep disturbance or insomnia. And these arrows are pointing to the subcategory. So anxiety is more of a subcategory of stress, and so you'll be able to see how the first arrow, sudden onset of intense fearlessness, that's categorized as a panic attack. And so PTSD, or post-traumatic stress disorder, that is a subcategory of stress. And so here at the bottom, the under the last circle, these human emotions can't really be modeled in anxiety. And so anxiety is more of a crippling response that hinders an individual unresponsive. And so this will be important when I'll be talking about the tests that were used to identify the receptors later in the discussion. And so just like depression, anxiety is also fairly easy to model in model organisms such as mice. And so anxiety is distinguishable from depression due to a specific provoking stimulus. And so here you're able to see how these symptoms can be modeled fairly easy in mice, such as this first circle, a panic attack, how this can be modeled in mice. This symptom modeled in mice increased flight from a predator. And so these at the bottom, uh, this last arrow, there are fewer limitations for anxiety compared to depression, but mice can still be used as model organisms for humans. And so here you'll be able to see the complexities of the human and mouse brain. So the human brain is more complex than the mouse brain, and you can tell here just because of the folds in the human brain compared to the mouse brain. Um, the mouse brain is a good model organism because there are many conserved structures. As you can see, the cerebral cortex in the mouse compared to the human, even though it is not as complex as the human brain, it is still a good model organism. So what exactly are the anxiety receptors that were identified by researchers? So the first one, the first major one, um, CRF1 and CRF2 receptors. These mediate neuroendocrine autonomic behavioral responses to stress. So these receptors are expressed in endocrine and immune cells. So endocrine cells are cells that secrete various hormones in the body, while immune cells are different types of white blood cells, which are often activated when there is an upregulation of anxiety symptoms. So here is a view of CRF1 and CRF2 receptor seen at the bottom, this bottom picture. And so these two receptors are implicated in physiological and behavioral responsivity to stress and emotions, including fear and anxiety. And so CRF coordinates these different responses to stress. And so this next one, 5-HT2 receptors, is different because it is a serotonin receptor that regulates stress management. And so researchers found that a downregulation of this 5-HT2 receptor is actually linked to have antidepressant effects, which is good because an overstimulus of this receptor causes hallucinations in an individual and seizures due to the rapid firing of the neurons in the brain. And so here is a view of what the receptor looks like in the brain. And so excessive transmission of this receptor actually causes symptoms of anxiety. So this was a novel find for researchers, which actually led them to this next receptor known as a Npy2 receptor, also known as a Neuropeptide Y Receptor. It is implicated in the pathophysiology of certain mood disorders, including depression and anxiety, which is why the researchers knew to knock out this receptor because of previous knowledge they had about this receptor. And so a downregulation of this receptor is actually linked to reduced stress and depression coping. And you can kind of see a trend right now that a downregulation of these receptors is linked to antidepressant effects. And so there was a study performed in 2003 that showed that the inactivation of this receptor in mice showed that there was increased motility during a forced swim test, longer time in a light-dark paradigm, and also increased mobility in an elevated plus maze. And so those different tests are going to be referenced later in the talk. But here, the NPY2 receptor, and box A on the left, this is the expression of the receptor, and then a knockout on the right, or inactivation of the receptor is seen on the right. So what is the main question of this research? And so my hypothesis for this research is that all of these receptors have been implicated to be upregulated, but we don't really know that if in their upregulated form, if they are active or not. And so I think that these three receptors are important linchpin modulators of stress response. But if that's the case and we do transgenic knockouts in each receptor individually in a mouse, we should expect to see reduced signs of stress and anxiety and depression in the mouse. And so how were researchers able to model anxiety in mice? And so that's what I'm going to talk about right now. So here are some experiments that were used to examine depression in mice. Some of the more popular tests that were used were the forced swim test and the tail suspension test, as well as the light-dark paradigm. And these are tests that are used where mice are exposed to trauma and stress, and then anxiety and depression are quantified using the tools that were mentioned previously. So here is the first test that I'll be talking about today. It's known as a forced swim test. And so what happens in this test is a mouse is placed in an inescapable cylinder filled with water. And so once the mouse is placed in the cylinder, um, what it first tries to do is frantically escape the from the cylinder. And so here is when the anxiety and depression receptors are measured using the tools that were previously mentioned earlier. And so what we'll see is that in these knockout studies, once the receptor is knocked out, the mice will show reduced motility in a forced swim test. And so next, you'll see in this tail suspension test, so it's a mouse is suspended from its tail from the top of a box. And so anxiety is reduced in mutant knockout mice in a tail suspension test. So again, these receptors were chosen, and so they were knocked out, and so there was reduced anxiety in these mice. And so, as I mentioned previously, anxiety is kind of a crippling response, and so once the mice has the receptor knocked out, they're able to freely move around in an enclosed space. So here is a final test, the light-dark paradigm. And so a mouse is placed in this box with the receptor first. And typically, the mice is going to want to stay in the dark environment. But once the receptor is removed, it's going to spend more time in the light, because mice don't like to spend any time in the light because that exposes them to uh, more predators like humans or different animals. And so the anxiety receptor tends to keep them in the dark and closed environment. But once that's removed, they tend to spend more time in the light, actually. And so here is kind of an overview of a knockout in the CRF1. And so here on the right, the major results of the knockout study. So here in the light dark box, you can see the results are an increased activity. So CRF1 is, like previously mentioned, it's more of an anxiety receptor. And so anxiety is a crippling response. And so that crippling response hinders the mouse in the same spot for a long time. And so once that's removed, it's like the mouse doesn't have any anxiety really. And so it can freely move around as if there's no predators around. And so the mice lacking the CRF1 receptor exhibited behavior suggestive of reduced anxiety. And so the mutant mice, or the knockout mice, exhibited increased locomotion. And so this plays a role in the expression of anxiety. And so you can see at the bottom here, the antisense oligonucleotide inhibit gene expression. So the defensive withdrawal, you can see that there was decreased latency to enter the open field and an increased open field time. So this was what was expected once the CRF1 receptor was actually knocked out. And so here are some conclusions of the test. CRF1 and Npy receptors saw an increase in locomotive response. The inactivation of this saw an increase in locomotive response, which was predicted or expected during the forced swim test and the light-dark paradigm. And so the 5-HT2 and Npy2 receptors showed a decrease in struggle time during the tail suspension test. So this is also expected because once those receptors were removed, it was like the mouse was able to be less anxious. And so first it was able to struggle, but once the receptor was removed, it showed a more calming effect, and so that's why there was a decrease in struggle time for the tail suspension test. So here is the take-home message. And so identifying these major receptors can help researchers target similar receptors to help upregulate or downregulate their expression in the body. And so these receptors are promising targets for gene therapy, which I'm actually interested in, which is what I would like to talk about in another discussion possibly, if I ever get the opportunity. And the inactivation of these genes can help us understand and treat anxiety disorders, which could be helpful if another pandemic or another worldwide global lockdown occurs. And so all three of these have been shown to be profoundly upregulated in their expression during a period of anxiety, which this goes back to my hypothesis about we don't really know how they act in their upregulated form. And so the downregulation of these receptors showed a decrease in anxiety, depression, and stress, which was expected. And so if you have any comments, please feel free to email me. And I look forward to hearing from all of you. Thank you.